This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 599, recorded on April 7th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here, and it's another gorgeous day here with partly cloudy skies, and I think it's up in the 60s today, but, just sitting around the house.
It is pretty nice out, yeah. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We got 83 Fahrenheit, 28 C, partly cloudy, a little on the warm side, but nothing like it's going to be down the road. We have another cold front coming through in a couple of days with some rain. It's all good. And I've already hit my 10,000 step goal today. So we're all good.
And from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is 64 degrees Fahrenheit and we have a little bit of sun today. And it's, more teaching on Zoom time.
You do that today?
I did. Yes. I have a class, at least one class every day.
Wow. It's a lot of classes. So you're teaching virology?
I am teaching virology. I'm teaching microbiology, and I am teaching a genetics lab.
Wow. All online.
All online.
Someday we'll have to find out how you do a genetics lab online.
Happily, we had just finished doing a whole bunch of cloning and making a whole bunch of constructs. Mm-hmm. And so we've been able to do bioinformatics ever since.
Today we have a guest here in, I'm at home in New Jersey, and my guest is right here with me. She is a pharmaceutical consultant. Doris Cully, welcome to TWiV.
Hello, everyone. Vincent's been trying to get me to do this for quite a while.
I was wondering if you'd been on the show before. I was hallucinating that maybe you had, but I guess that's not true.
No,
but
I had a long commute.
Yes. Yeah, it's true. Well, we have a good reason to get you on today, and I know you're only here for 25 minutes or so, so let's get right to it. And it is a paper, it's a preprint. Is it a preprint? No, it was published, actually.
Now, yeah.
It's published it's in Antiviral Research, and the paper is called "The FDA-Approved Drug Ivermectin Inhibits the Replication of SARS-CoV-2 in Vitro." This comes from an Australian lab in Victoria. The Royal Melbourne Hospital at the Peter Doherty Institute, where I've been, and also at Monash University. And Doris, tell us about your experience with ivermectin, just as kind of a background. Why would you be a good person to talk about this paper?
I worked on ivermectin at Merck for many years when it was first discovered, and I was involved with identifying the mechanism of action, which we use xenopistosized expression system to identify invertebrate class of receptors that had not been identified before, called the glutamate gated chloride channels. So I was asked to look into this paper by some friends of ours that work at Mount Sinai recently, and so that's why I did some background sleuthing. Mm-hmm. But I have no concept at all about how it's working in viruses.
I must remind two things I wanted to say before we get into the paper. First of all, you did your PhD on phages, right? So you kind of have some virology background many years ago.
What phage, Doris?
Phe105, gram-positive bacteria, lysogenic bacteriophage.
Cool.
So B. subtilis.
Yeah, ironically, years later, I ended up working on MRSA at Merck for a while, too.
Rich did work on T4?
T7.
T7. Yeah, we use that a bit, too.
The other connection here is that Brianne Barker is a professor at Drew, which is where your old friend Bill
Campbell went.
Has some kind of honorary appointment.
He's retired now.
Yeah.
Yes, we have a whole area of retired pharmaceutical scientists who have labs and who mentor undergraduates.
Right. So his lab was right next to mine.
I just remembered. Oh, sorry. Go ahead.
That's okay. His office was next to mine. So some of the stories about ivermectin, well, I won't get into it right now, but maybe later in terms of its safety, were from conversations with him.
You had an office next to him.
Yeah.
That's
Oh,
cool.
wow.
My lab was next to him.
Cool.
That's cool. That's very cool.
And I just remembered where I met Doris. It was you came to Columbia and gave a talk about ivermectin and river blindness and your work at Merck.
Oh, yes, I remember that.
That was
With
Lex.
Lex van der Ploeg?
Yes,
Yeah,
that's right. Yeah.
Yes.
It was like a careers series, I think.
Yeah.
So I knew Lex because I used to work under Panasomes
Right.
Right.
at Rockefeller. So
Okay. Well, this paper says that ivermectin inhibits SARS-CoV-2. And let me just tell you the virology here. There are essentially two experiments where they infect cells in culture, and they use
they do a dose titration of ivermectin.
they do vero cells, African, or I should say vervet monkey
cells.
kidney cells, and they add the drug at the time of infection first, and they show that it really brings down virus yields when measured by PCR actually. And then they do a dose titration, right, where they've added the drug, I think, a couple of hours after infection. So let's start with that. So they say, Doris, the IC50 is like 2 micromolar roughly.
Right.
So maybe you could tell us what that means.
So the question, I hate the title because I will, I'm sorry to the authors, but they say FDA approved, but it's not approved for any antiviral work. It's approved for antiemetic use and also for treating, for veterinary use for treating invertebrate infections such as mites and ticks. And many people who have dogs have used ivermectin as heart guard, and also cattle people have, for ascaris, etc. So it was used in humans to treat river blindness originally, and subsequently to treat other parasitic nematode infections in East Asia. But its dose is extremely low compared to what the IC50 is for viral inhibition in cell culture. It was the invertebrate target is sub-nanomolar KD, and the IC50 from this paper for inhibiting virus replication is in the 1 to 2 micromolar range. So that is why I don't particularly care for the title, because it's inferring that it's FDA approved for viral work, and it's not. So, the other thing that's mentioned in the paper is the potential for use in other viral infections, and they reference a phase 3 study that was done in Thailand for dengue virus. And if you look at the publications in vitro with dengue virus for the 4 different serotypes, which they reference in the COVID paper, the IC50 is similar to what this paper is describing in culture. It's about 1 to 2 micromolar IC50 as well in cell culture. And when they used the recommended approved FDA dose in the phase 3 study in Thailand, they did not see clinical benefit and decrease, no change in viremia. So, clearly using the approved dose is not efficacious. It translates also to the fact that it's much less potent against viruses.
So this is for the non-science folks in the audience, the micromolar versus nanomolar. This is 1,000-fold difference in concentration we're talking about, right?
It's actually more like 10 to 20,000-fold more.
Right.
Because it's much less potent on the viral target in cell culture as compared to binding assays in vitro for ivermectin on nematode targets.
So this is probably not a concentration that you could get in a patient without really serious problems, right?
Well, we don't know that. All right. So
Okay.
part of the problem is because it's so efficacious, it was never really dose titrated in safety studies to those levels. I've been trying to find literature about that. And years ago, when this was developed, it was really for veterinary use. And there's a sparsity of data that you would normally see with a drug developed for human use today for, for this compound. It's been in hundreds of millions of doses in patients throughout the world at these low doses, and really very little, if any, adverse effects. That being said, originally it was given for river blindness once a yearly dose. So the other complication is it never was really dose titrated to these high levels in safety studies. There's some exceptions, which I'll get to. But the other thing is it wasn't given multiple days of dosing. So the current approved doses in the FDA approved doses are 0.1 and 0.2 mg per kg. If you wanted to try and achieve a micromolar concentration, you'd probably have to give like 25 mg per kg. It's a lot more. So the other complication is it's only approved for one unusual indication for scabies for once every 2 weeks of dosing, but it is used more frequently for that than that in the clinic. For scabies, it's like once every other day dosing, but again, these very low doses. So it is possible that you might be able to get efficacy at a higher dose. There are toxicities associated with high levels of dosing in animals. But there are some reports that people in the veterinary field have overdosed themselves, either intentionally or by mistake, and really haven't had very major symptoms. The major route of toxicity of ivermectin is the GABA receptor in the central nervous system. And ivermectin is a substrate for the blood-brain barrier pump, PGP, or multi-drug resistant receptor. So it is pumped out of the brain. And we know that if you don't do that, it's toxic because there were inbred strains of collies that were succumbed to ivermectin at even these low doses. And when the PGP protein was sequenced from those collies, it was found they had a mutation. So they weren't effectively pumping it out of the brain.
Wow. So what's in the targets for which it is approved? The protozoans or invertebrates?
No. Sorry, go ahead.
I want to know what the drug target is in those organisms.
It's the glutamate-gated chloride channel that I worked on. It's a novel channel that is similar to the ligand-gated ion channel families of GABA and glycine receptors. So the GABA receptor is so if you're interested in molecular evolution, which I am, invertebrates do not have chloride channels that are gated by glycine. So they use glutamate both for inhibitory and excitatory neurotransmission. So it seems that and vertebrates, which we belong to, some of us, we use GABA and glycine for inhibitory neurotransmission. So if you look, if we had the genome at the time, we would have probably just cloned anything that was homologous to GABA receptor and found it, but we didn't at the time. So we used C. elegans as our strain is very sensitive to ivermectin to make expression libraries and do expression cloning in xenopus.
So in the invertebrates, is the cause of death a neurotoxicity?
So it results in paralysis. It doesn't actually cause death. So ultimately paralysis will cause death. But one of the problems with treating river blindness with other antiemetic agents is it would cause death of the circulating filarid that would then die and deposit behind the eye as well. So, one of the things that was good about ivermectin is it, I guess, allowed for time for clearance as opposed to just sudden disruption of the cells and immune response.
So the way that this drug is hypothesized to work against viruses in this paper is completely different than what it's thought to be doing in those infections.
Yes.
Yeah, they suggest that it's blocking nuclear import of something, right?
Right.
And that's funny because the virus, as far as I know, no coronavirus protein is imported in the nucleus. So it would have to be a host target. It would have to be a host protein that's going in. Maybe, Brianne, it could be a interferon transcriptional activator or something like that.
Well,
Perhaps,
let me talk about ivermectin a bit. Ivermectin is a natural product made by actinomyces found in the soil. So I've always, again, back to molecular evolution, actinomyces probably makes ivermectin to compete with other things in the cell, including nematodes like C. elegans. So it also has antifungal activity, but it is a very large compound. It's almost 900 molecular weight. So to me, this is not some small enzyme pocket inhibitor.
Mm-hmm.
This has got to be something that's, it has an affinity for chloride channel pockets. It's extremely hydrophobic. It loves to partition in membranes. So, and the papers do not describe any cellular toxicity. So, it's curious as to if it is disrupting something that's a membrane or very hydrophobic type of a pocket that's in the cell. It's not something that if you disrupt it, it causes cell toxicity.
Yeah, they show their picture, ivermectin binding to importins, which are nuclear import proteins in the cytosol, and blocking import. But I don't think there's any
mystery purple protein.
Right.
Yeah, but as I said, I'm not aware of any coronavirus protein, excuse me, that has to get in the nucleus. So that's a weird mechanism. But Doris, the dosing is the issue here because, as you said, the dose that's FDA approved is not enough to inhibit this virus, according to the graphs they've presented, right?
Yeah, they do know. I think mice are probably the most sensitive species.
Mm-hmm.
Whether or not that translates to man or not, it doesn't really matter because that's what the FDA would use for minimal tolerated dose allowed.
Right.
So there are, if you give high doses like 25 mg per kg in mice, you do get some lethality.
Mm-hmm.
So that's the only data I could actually find. There's some data that's slightly lower than that, but again, one would have to do repeat dose studies at lower doses.
So even though it's FDA approved at lower doses for its indicated uses, if given what we're seeing here, you'd probably have to go through most of the FDA approval stuff all over again to get it approved at the higher doses, right?
Well, you can't just jump in at a higher dose.
Right.
But I would, start at the low doses at multiple day dosing because it is going to accumulate probably in tissue because of the hydrophobicity.
Right.
So if you were going to design a trial for SARS-CoV-2, how would you do it?
Well, I'd take healthy individuals, not infected, and start with the safe dose and go up. But you need some animal data first to see what your no effect level is. You'd have to do dose titration, single dose, and then multiple day dose titration.
In mice, you would do that?
Probably rats.
Mm-hmm. And so you couldn't do that before you went to the FDA for ivermectin for a viral infection, right?
You could not do that? You could.
You'd have to do that before the FDA would even look at it, right?
The FDA might have access to data that I can't see online.
Okay.
So these, many of the studies were done early 80s, mid 80s, and they might have some of the paper trails because none of it is electronic.
Yeah.
Right.
So there are some newer indications that Merck was not involved with that I found, some information where they did genotoxicity. So looking for 2-year studies for cancer. And that was for topical applications which is completely unrelated to parasitic infections, probably because of antifungal activity. And that study did dose up to 9 mg per kg, but it's only in the label. So there's no data. There's no clear data. They have the data at the FDA.
So assuming they have the data, the FDA has that, you could then find a dose. Would you still do a multi-dose trial and see if it prevents infection?
Because of the situation right now, I don't know if they would have to do a full trial.
Mm-hmm. You had some concern about treating infected patients.
I don't know enough about what's going to happen to the blood-brain barrier in these patients.
Mm-hmm.
I put that question out to clinicians. But, if the blood-brain barrier is perturbed.
In infected people.
Yeah. I mean, the label of ivermectin also, points out a couple of other issues. Co-dosing with compounds that act at the GABA receptor is an issue. But in particular, other than those items, no, I don't think there's an issue. Again, when you get into this dose range, there's a lot unknown about it.
So do you think someone's going to do a trial with these low doses and find that it doesn't work? The doses that are approved in
That's all they can do right now.
Right.
In the U.S., right?
So doses slightly higher have been used outside the United States, but really just slightly, 0.4 as opposed to 0.2 mg per kg. It's really not much more. I think the Zika study in mice went to 4 mg per kg, and they did not have lethality, but they only dosed 4 days, and they refer to that in this publication as well.
And did that study in mice look at whether it had any effect on virus at that concentration?
They say efficacy. So I think they're scoring. I can't remember now. I looked at that paper quickly. The other thing was they did IP injection. So it's hard to correlate that. They might have actually had much higher exposure than the oral, because I think there's some...
The drug, you mean? They put the drug IP?
Yeah.
They must have mixed it with virus probably, right?
No.
No?
No. I mean, so they looked, I think, at the same endpoints that these other publications should, my guess would be, but I don't remember offhand.
Okay.
It's reference 14.
In their paper, yeah.
That paper.
Sounds like it's not a promising candidate, despite the nice virus inhibition curves, right? Yeah. The other thing, one more thing. This virus predominantly replicates in the respiratory and gastrointestinal mucosa. So will the drug get there at sufficient quantities to inhibit virus replication?
I mean, I don't see any reason why it won't. It's in the bloodstream. It's partitioning to all tissue.
Okay.
It's not, drugs don't have particular tissue tropism in terms of where they're going necessarily, unless it's cleared somewhere.
So drugs typically go everywhere.
Well, if it's carried through the plasma, yeah. I mean, except for the brain.
But ivermectin's oral administered in people?
Oh, yeah.
Okay.
Actually, that's the only formulation that's approved for humans. It is tablets. There are topicals that are now being used for like rosacea or lice, but it's not
Got it.
And it's not amenable to IV. So, because of the physical chemical characteristics.
I didn't know that I thought you had to do bioavailability studies on any drug to make sure it gets to where you want it to go.
Well, bioavailability is just the percent that absorbed to the gut that gets into the plasma.
Okay. Got it.
I mean, you give a drug IV to see 100%, and then you give it oral, and then you just look at you calculate the amount absorbed.
Okay. Right. I didn't know that. So you don't need, in any drug study, you don't need to see what tissues it goes into in the animal?
Well, I mean, it depends on a lot of things. So if it's cleared very rapidly through the liver, then, you're going to have greater concentrations potentially in the liver. So, it depends on.
Yeah.
But ivermectin probably has, relatively high volume of distribution, which means it's partitioning to tissue.
Yeah. What does a volume distribution mean for a drug?
In my mind, because I'm not a, pharmacokineticist, is just I use it to determine how long a drug stays in tissue.
So it's pretty high for ivermectin, and that's why they could dose for river blindness just once a year.
No, that is a mystery that still hasn't been solved. The drug does not hang around for a year.
Okay.
Well, I haven't read this paper in detail, but I've just been skimming through it while we're talking, and the authors don't make any mention of this issue with the difference in dosing between its prescribed use and this use. So I really appreciate your insights, Doris, because that puts a whole different spin on this.
Yes.
Yeah, that's really interesting.
Well, they're probably virologists, right? And thought, let's try this, but they don't know anything about drug availability, etc.
Yeah, I'm sitting here looking at the references that I want to read later about how ivermectin may influence nuclear localization.
Yeah.
Seems like they have a couple of potentially interesting papers I might like to read.
Doris, your time's up. Anything else, guys, before she leaves? Is that it?
I'm good.
Okay,
come back on the show
Doris.
sometime.
Thanks, Doris.
Yes, come back. We love to have you. Thank you.
All right, take care.
Bye.
Thank you.
See ya. Well, that's interesting.
Yeah.
Shit I wouldn't know, right?
No, I wouldn't know that.
That's why you bring on an expert when she's in the same house.
I have to say that, although it's where you start, I'm usually very wary of studies of inhibition in cell culture. I mean, mice lie and monkeys exaggerate, and cells in culture, I don't know, but they're, worse than liars. Okay.
Well, you have
to start. You have to start there. That's where you have to try. But just because something works in cell culture, that's a long, long way from efficacy in people.
Actually, someone pointed out the same issue with hydroxychloroquine, right? It works in cell culture. Someone pointed out it works against a number of viruses in cell culture, but doesn't always help in people.
Right.
So that's important. I can see where her issue with the title, the FDA-approved drug ivermectin inhibits replication of SARS-CoV-2. Yeah, FDA approved for what, right?
The title is technically correct, but it is misleading.
Yeah.
Yeah. Okay, let's do a couple of other things before we hit the email. Thanks everyone for joining us on this unusual Tuesday TWiV. Well, maybe not so unusual these days, right?
All right.
One story I wanted to cover is related to something we talked about last Friday. A tiger at New York's Bronx Zoo tests positive for coronavirus. And I like this story because the name of this Malayan tiger is Nadia, which of course is our daughter's name. So this tiger got sick, developed a cough, and so they, did x-rays. It's not easy to check a tiger. You got to sedate them, I guess, right?
Right.
I'm not going to stick a swab up a tiger's nose when they're not sedated.
Yeah, I think that it notes here that there were 3 other tigers and 3 lions who also were sick, but they only tested Nadia because it was too hard to test 7 big cats.
And they thought at some point, let's try SARS-CoV-2, and she was positive.
I'll be darned.
But they think they're all going to be fine. I don't know. I guess if you're used to tigers and lions, you can figure that out, right?
Yeah, you could judge how bad their disease is. But I didn't know that they'd actually all had symptoms. I guess that makes sense. A dry cough. So
I wonder what a tiger or lion sounds like coughing. I never heard one, right?
Probably like a big version of a house cat coughing up something, I guess.
Yeah.
I've certainly heard house cats coughing.
That's true.
Yeah.
So I've never been to the Bronx Zoo. You've probably been to the Bronx Zoo.
Oh, neat. Nice zoo.
Yeah.
Yeah.
The last-
you've probably even seen Nadia.
I don't know. The last time I was there, we actually did a TWiP with Paul Calley, who is the head of the New York Wildlife something or other. What's
Is
the Wildlife Conservation Society, which basically runs all the zoos in New York.
Is the
He's the head
Bronx
of-
Zoo where they noticed that the crows were dying in the West
That's
Nile?
absolutely right. And
It is.
he took us behind the scenes at the zoo. So we went early one morning, me and Dixon and Daniel Griffin. And he showed us, you see these freezers? We have all samples of dead birds in there from West Nile. They got all kinds of stuff. It's a gold mine. He said he loves to collaborate with university scientists. If you have a good project, he's willing to do it. Anyway, he's quoted in this article. I thought that was pretty. He's the chief vet at the zoo. And they say they think an asymptomatic employee might have infected them, which I guess makes sense, right?
Sure.
Sure.
Mm-hmm.
Anyway, so that's
But
in line with the...
That kind of highlights how easily transmitted this thing is, right?
Mhm.
Because I'm guessing that at the Bronx Zoo, you're not going to be up in the face of the tigers, even if you're a zookeeper, right? Unless you're not planning on being there for very long. So I guess this was an asymptomatic keeper who was in the general vicinity, and
I guess you could get within 6 feet of a
sure,
big cat, right?
Yeah.
if it's
But
in
remember-
a cage
Maybe
and you're
if you're
cleaning
feeding
outside
it.
the cage or you're feeding it, I guess, yeah.
I'm not
Also-
sure I'm getting within 6 feet of a yeah, if there's bars in between.
If there are bars in between, yeah.
But it could also be contact, right? So the keeper could have contaminated something in the cage.
Sure.
And the lion, tiger licked it up, whatever. So all kinds
Coughed on
of
the meat,
scenarios.
yeah.
Yeah. This data, as well as some other data about infection of cats,
Yep.
has made me think a lot about our conversation with Ralph Barrett
Yes.
and the idea of a reservoir being established.
Oh, this is so real. Yes, absolutely.
Spillback.
Spill.
Right.
Mm-hmm.
So yeah, we did the paper last week where it was a lab showed that you could infect in a lab cats and dogs and ferrets.
No, not dogs. Cats and
No
ferrets. Dogs were okay.
Dogs were not infectable,
Right.
but there were a couple of reports out of Hong Kong of dogs being positive. I don't know if that's real or not. Anyway, let me read this letter from Paulo, who is he is a veterinary professor in Brazil, University of São Paulo. He writes, you might remember me. I'm a virologist professor at the Veterinary College, University of São Paulo, Brazil. One of the guests on the first TWiV in Brazil in 2011. I talked a little about my work on avian coronas. In episode 598, you discussed a manuscript on susceptibility of cats to SARS-CoV-2 infection. This manuscript is causing major issues in the veterinary community here for pet owners, and its cats are being abandoned.
I was worried about that, man. What a bummer.
And some private vet labs are already selling SARS-CoV-2 detection for companion animals. He writes, the manuscript has plenty of flaws, which I summarized in a letter to the editor for Nature. And he puts a copy of the letter below, in which he basically says they only used PCR, so we don't actually know if they are shedding infectious virus, right? Or it's even replicating. He's not, which I would say is a fair criticism, right?
Yeah, absolutely.
Virus transmission to a sentinel cannot be promptly interpreted as active infection, as the inoculum, itself may roll from animal to animal, and this could also result in antibodies and virus re-isolation.
And he points out cats are known to be ubiquitously infected by feline coronavirus.
Indeed. Host group studies are invaluable to guide control measures, but precipitated news might lead to the election of scapegoats. That's a good one. I like that. Huh. And in fact, we also had a letter from Vamsi who wanted to know about infection in cats and dogs. If cats and dogs are infected and show no signs, could they become another reservoir? Yeah.
Yes.
For sure.
Mm-hmm.
And, well after this outbreak ends, your cats could pass it back to an uninfected person, right? It's kind of a nightmare situation, but don't abandon your pets.
That's what's going on.
If it's what's going on. We don't know. We should say, and don't abandon your pets, please.
No, gosh, don't abandon your pets.
No.
Not only is does that make somebody a horrible person for doing that, but it also, but if this is the case and if this is going on, then your pets should be locked up in your house so they don't infect anybody else.
Yeah. That's sad.
It did. I did think when I saw this about the, potential of cat susceptibility, this might be something to look at as a possible intermediate host in the original spillover.
Mm-hmm.
Yeah, absolutely.
Because we now know it probably didn't originate in the wet market in Wuhan. The initial cases predated that, didn't have contact with that market. And then the theory is maybe somebody like a guano harvester got it from bats or got it through an intermediate species to bats. But cats will certainly hunt bats. So,
yeah,
I don't know.
Sure. It's possible. We'll see. Ongoing. All right. The other story I wanted to bring up. This is a paper out of University Hospital Munich. It is a preprint at medRxiv. Level of IL-6 predicts respiratory failure in hospitalized symptomatic COVID-19 patients. They looked at 40 patients in February, from February through March, and they found that elevated IL-6 was strongly associated with the need for mechanical ventilation. And maximal IL-6 level by each patient during disease predicted respiratory failure with high accuracy. So if you remember, Daniel spoke on Friday about using the monoclonal against the IL-6 receptor to treat some of his patients. He thought it actually was working well. And so this is in support of that, right?
Right.
Yeah.
Yeah. This sort of goes along with the idea that maybe sort of excess cytokines or cytokine storm might be part of the disease pathogenesis.
Right.
Right. In particular in the acute cases. Or the
severe cases.
The severe cases.
And you pointed out last week on Immune, Brianne, that IL-6 is probably not the only culprit, right?
Probably not. IL-6 is probably a relatively easy cytokine to measure. And so usually we see kind of a whole array of inflammatory cytokines being made at the same time. So it's not just IL-6. This may be just the one that we're measuring best here.
So this monoclonal to the IL-6 receptor, right?
Yep.
And that's an approved drug, correct?
It is, yeah.
It is.
And so I'm wondering what that was developed for. Does anybody know? Any of us know?
I think Brianne knows.
Brianne is Googling right now.
There's a name. We have the name before. What is it?
And it starts with a T. Tocilizumab.
Right.
Whoa.
So what's that? What is it for? Immunosuppressive drug, mainly for rheumatoid arthritis.
Ah, okay.
Okay,
So inflammatory disease of one sort or another. Okay.
So I think we'll see more trials of this because this was relatively small, and therapies as well would be interesting if that could be a predictor of whether you're going to go into the ICU or not. That way, I don't know what you bring a patient in, they have high IL-6. What do you do? I'm not a doc, so I have no idea.
Yeah.
I guess you start treating them with that IL-6 inhibitor.
Maybe. So, Brian, the idea here is that some people, for reasons unknown to us, when they get infected, they just make uncontrolled IL-6 and other pro-inflammatory cytokines. Is that correct?
That is correct. And that could be due to some aspect of their genetics or co-infections or previous infections. Lots of things could be going on to make that change in IL-6.
Right.
Okay.
There's actually been some really interesting data also about innate immune responses and cytokines with a vaccine BCG. And so, in fact, I have seen all sorts of things that have made me think a lot about inflammatory cytokines recently, along with COVID.
I think we have some letters about BCG coming up in the queue.
Okay, we'll hold back. I wanted to talk about that, but we'll wait for the letters. And I just want to warn everyone, I don't know if I mentioned this on Friday. Remind me if I did. There are some studies that show that oral polio vaccine actually inhibits other infections for probably the same reason. So don't be surprised if you see in the next few weeks it being suggested to give people OPV.
Right when the global polio eradication campaign has been suspended because of COVID-19.
Yeah, maybe we could resume it for COVID.
Yeah, resume it for COVID-19.
And then to get two things at once, right? That would be cool. All right, let's do some email here. Brianne, can you... Let's see, we're down to a non...
Rich's
Oh,
blog
sorry,
post?
yes. Please, Rich, you wrote a nice blog post. It's at Virology Blog. Yeah, why don't you review for us what you're thinking there? Thank you.
Okay, and I first of all appreciate you posting this on your blog, because that comes with it comes with an audience, which is great. So we have talked a lot about what's going to happen when the restrictions are lifted. Is there going to be a second wave? How big is the second wave going to be? And et cetera. And in my mind, in order to try and understand that, we need to know how many, after the first wave is over, how many people have actually been infected, so that we know sort of the density of the susceptible population. And that's a number that I've had a hard time finding. And in looking over this site, so by the way, the title of this blog post is Infection Fatality Rate, a Critical Missing Piece for Managing COVID-19. And in fact, as I think back on it, what I'm really looking for here is infection rate. How many people are infected at any given time? Or have been infected, not just active infections, but have been. How many people are presumed to be immune because they have antibodies? So at any rate, in order to try and understand this idea of the residual susceptible population after the first wave is done, I've been looking around for numbers, which are hard to come by. And one I came across was from this modeling site from the Institute for Health Metrics and Evaluation, which we discussed this site some time ago, and it's become a kind of a go-to site for these sorts of predictions. And what their mission is to model the impact on hospitals and whether or not there are going to be enough hospital beds. But as sort of a sidelight to that, they can also, part of their modeling, they're modeling deaths per day over time and making predictions as to what this first wave is going to look like. And one of the spinoffs from that is, after this levels off, how many people they predict will have died, which at the time that I wrote the blog, they figured after the first wave in the U.S., there would be on the order of 93,000 deaths total. They've downgraded that now recently to about 82,000 with revised figures. And I looked through the frequently asked questions in this site, and one of the quotes I ran across was this: by the end of the first wave of the epidemic, an estimated 97% of the population of the United States will still be susceptible to the disease. So avoiding reintroduction of COVID-19 through mass screening, contact tracing, and quarantine will be essential to avoid a second wave. And I thought, holy cow, you mean after all of this, we'll only have 3% of the population infected? And I don't believe that the virus is going to be gone, right? Like beaten back, but now broadly distributed through the population and maybe thinly distributed and poised to pounce on us once we lift the restrictions. And if 97% of the population are susceptible, I don't see how the second wave is going to be any different than the first wave, except perhaps that we're a little more ready for it in terms of hospital care. So I wanted to know kind of where they got that number. So I actually wrote them, but they're busy. But you can kind of figure it out, because if you say that they're predicting at the time 93,000 deaths, and the population of the U.S. is 330 million, then that let me see. How did I go? Then you can. I'm losing my train here.
You're doing well.
At any rate, I went through the calculations. And what I came up with was that they were probably using a 1% case fatality rate, okay, to figure out that 97%, okay? And so the 1% case fatality rate, that's a case fatality rate. And as we've discussed here before, that's different. That doesn't count all the infections. It just counts the people who were met the appropriate indications for getting tested and who were fortunate enough to have testing available. And it's probably only a small fraction of the infections. So what you really want to know is the infection fatality rate or the infection rate. And that number, we don't know. There are no data at this point. You can find sites that try and figure it out. I found a site called the Center for Evidence-Based Medicine. That site actually, they go through all of the scenarios that we've talked about, all the places you can collect data to try and figure out case fatality rates or infection fatality rates. And they come up with a global estimate of a case fatality rate of 0.51% and an estimate of the infection fatality rate at 0.1% to 0.26%. Now, that has all kinds of caveats. And as I said, it's a kind of a made up number because we actually have no data whatsoever on infection fatality rate. However, if the infection fatality rate is really 0.1%, then by my calculation, after the first wave is over, there may be as much as 30% of the population that is resistant. And that's a much bigger number. To me, a virus trying to come back with 30% of the population immune, the second wave may have significantly different dynamics than the first wave.
And we should bear in mind that those infections will not be evenly distributed across the U.S.
Right. Yes.
Right.
So this got me thinking. We can get a number for the infection rate or the infection fatality rate, because as we've already discussed on the show, kits have become available recently that actually simply adapt an existing technology to SARS-CoV-2 that can measure antibodies in the blood. And these kits are very simple to use. You just take I've never actually seen one, but I imagine they're maybe about a plastic thing that's maybe about an inch wide and 3 inches long. It has a well at one end and a little elongated window at the other end. And you mix a drop of blood with some diluent and stick it in the well at one end. And then just like a pregnancy test, you look for lines to appear in the window, and the lines will tell you whether you've got antibodies to SARS-CoV-2. And they'll even tell you whether you've got IgM or IgG antibodies. So they tell you whether or not you've been infected. And if you have been infected, whether or not the infection was recent. They don't tell you whether or not you got an active infection. But that's not the question. The question is, have you ever been infected and are you immune? And
now those come with important caveats too.
Okay.
So one that there was just a news story about this. Apparently a lot of the tests the UK just got, millions of tests, are no good.
Not good. Okay.
A lot of this stuff is being developed very hastily, and I think that story is not going to be unique, that people are going to get these serology tests because they're adapting an existing technology. A whole bunch of companies of all sizes can throw these things together and say, hey, we've got a SARS-CoV-2 test, let's sell it. And I'm not blaming the companies for trying to get on top of this, but I think some of the quality control that would normally go into validating these things may not be quite on point. The other thing is with antibody tests, I mean, the example you just gave is a well-established test that's been well validated. Then you can put it into this sort of consumer grade product, and that's where you get things like the rapid HIV tests that are available now. But for something like this, I think what we're going to be dealing with for some time until all the bugs get ironed out is tests where you get much more ambivalent results. Like what dilution of serum do you get that gives you a reaction, and well, is this patient positive or negative, and we're going to set a cutoff point, but that cutoff point is not perfect.
Right.
And then there's a whole statistical game you get into where if you've got a test that's 95% sensitive, but only a small percentage of the population is infected, then you're actually going to get a lot more false positives than you would realize. I'm not going to go into all that, but it is important to realize that while I agree we need these serology tests and they're going to be critical in tracking this disease in the future, and even right now, we also need to take them with a grain of salt as they're being rolled out.
Right. Make sure it's done right. So at any rate, my bottom line is that I would like to see these tests because, ideally, if the bugs are worked out, ideally they can be done very simply by people who are not necessarily professionals. You could set up test sites. You could make them available at pharmacies and do the tests. And I would think that people would flock to these things because they would want to know whether or not they've been infected.
Yes.
And I would also create a database, all right, where if you get tested, whether you're positive or negative, and at least a zip code gets put into a database so that we know I want to score both the positives and the negatives. Okay, just the positives don't do you any good. You got to know the whole population: who's positive, who's negative, a date, and at least a zip code. That way, we'll get a geographic distribution as well, which is going to be important in a reemergence. And I can't believe that this isn't being considered.
Yeah.
Oh, it certainly is. Wouldn't these also be much easier because they would require blood instead of that deep nasopharyngeal swab?
Yes.
That people are having so much trouble with?
I would think so, yes. Just a prick on the end of your finger to get a drop of blood.
On top of all of this, I think the government should pay for it.
Yes, absolutely.
Yes.
Yeah.
This should be free. It is absolutely the case that we're still going to need the virus test because we want to detect these infections as early as possible. We need the PCR test to keep going, and we need those to be made as reliable as possible. And both of these types of tests are going to be critical to reopening things because we need to be on top of this as stuff reopens and not just wait for people to start showing up in the ICU to say, oh gosh, it seems to have come back again. We need to detect these cases very fast and do contact tracing and all the stuff that the successfully managed countries like South Korea and Germany did early and managed to grab control at the start. That's what we need to do for wave 2 is really get a handle on when an infection occurs and then quarantine everybody who's had contact until you figure out what their status is and just start putting out these little fires as they get going.
So, Rich, you want a plan?
Yeah.
Yeah. Well,
yeah, I want to actually have a plan. Yes.
Good luck.
That would be nice. I know. I know. I just, going back here, I can recover where I had my brain fart. If they estimate 3% of the population is resistant to the disease after the first wave, that implies in a population of 330 million people, just short of 10 million infections. And if there's 93,000 deaths and 10 million infections, then that implies to me that they were using an infection fatality rate of about 0.9%. So my guess is that they were using the 1% case fatality rate that's used globally. So I hope that the actual infection fatality rate is very, very, very much lower than that, and that there's a lot more people immune. But I think that that's probably a false hope. When I look at what's going on around me and in Austin, especially with the social distancing, I think when, in a month or so, when the first wave is sort of looks like it's over, I think that the exposure level, at least around me, is probably going to be pretty low. And it's amazing what the social distancing does. Just, this is anecdotal stuff, but there's a story locally about, I think it's probably hit the national news, about a group of like 200 University of Texas students that went to San Cabo for spring break in Mexico and came back, and everybody was going, "You idiots, what did you do that for?" And some of them were sick, and I think they tested all of them, and a quarter of them were positive. 50 out of 200 were positive. So that just goes to show you that if you get together a group of people where there is a possibility of infection, it can really spread dramatically. So that's a great example that makes perfect sense that the social distancing is necessary and gives you a feeling for what it would be like without it. But all of that same stuff is going to apply several months down the road if we lift the restrictions and there's a big susceptible population.
But wait a minute now. You're saying two different things. You're saying we need the social distance, but on the other hand, it's keeping the herd immunity down.
Yeah.
So what do you want to do?
I don't
know.
know.
Well, this has been the dilemma since the beginning, because the whole flatten the curve argument was that you want to spread out the infections to prevent overwhelming the hospitals.
Right. Exactly.
And the downside of spreading out the infections is that the total number of people infected over a period of time is going to be a lot less. And so ultimately, if the curve starts coming down, we probably will have exposed a lot fewer people, and that's good because we've got fewer dead people. But it's bad in the sense of herd immunity, which we probably won't establish as reliably at the end of that, and then people are going to say, "Oh, the curve's coming down. Let's reopen things." The one fortunate thing here for the U.S., as horrible as our response has been, is that it got going here later than a lot of other places. So we can look to China and Italy as examples of places where they're now dealing with this and deciding how and when to open things, and especially in China, I think we're going to start getting some good data on how far this thing really spread if they actually go and do the serology. I mean, I trust the scientists there to do that, but I think the government there is maybe a little reluctant to do that because of what it might show. In Italy, I think there's going to be a much bigger push to do that sort of thing and figure out what's happening as they move to reopen things. And I'm hoping we can get those data before we go and say, okay, let's all, open our stores again.
Yeah, because this has big implications both for, whether we might see a second wave or how many waves we would see, and makes it all the more important that vaccine research is going on so that we can increase that herd immunity someday down the line.
So a couple of assumptions that I am making all the time in this is one, that when the first wave is over, the virus is not actually gone. Okay. But it's lurking out there. And, I think that's probably true, but I don't have any evidence one way or another. The other assumption I'm making is that ultimately this is creeping towards this will creep towards a situation where we will have herd immunity and the majority of the population will be infected. I'm not, I have no evidence for that either, other than looking at the existing human coronaviruses. So those are pretty big assumptions. I would also say that it's interesting to look. There was I think it was Kathy's, a link that was in one of the previous shows to the National Geographic article that looked at the impact of social distancing on the 1918 flu in various cities. Mm-hmm. And now it looked to me like I don't see any situation in there where the second wave, there were some cases where the second wave looked like it was as big as the first wave. But there was none where the second wave was bigger than the first wave. I don't know that the times went out long enough, but I don't recall seeing third waves and et cetera. So at least in the case of flu, the second wave was, so after the second wave, it was mostly over, it seems to me. The second wave was not any worse usually than the first wave. But of course, that's flu. That's a whole different kettle of fish. You got seasonality creeping in, other issues.
I want to clarify two things. First of all, when you say lurking, there's nothing fancy going on there. It's just infecting someone, but it's a very few people and you don't notice it.
Right.
It's like flu in the summer. There's always virus around, but nobody notices because they're pretty rare. And then the other thing, people misinterpret this idea of a second wave. The virus is not changing.
Right.
It's just the virus has come back and there's a lot of population non-immunity, and then maybe it's the fall or winter where it's good conditions for transmission, and that just precipitates an outbreak because population immunity is so low.
This is why we need lots and lots of reliable tests available.
Yes, very true.
And a plan.
Yes, and a plan. We need a plan. I nominate Tony Fauci to come up with the plan.
Ooh, good idea.
I'm good with that. Did you see they are now making Tony Fauci bobbleheads?
Yes.
Good. I want one.
There's all kinds of Fauci stuff. T-shirts, bobbleheads.
Well, I just want to say we here on TWiV were into Tony Fauci before a lot of people. So,
oh, yes, for sure. True. But we were into viruses before a lot of people too, right?
That's true.
But welcome everyone and stay.
Yes.
It's a good place. We like you.
Viruses are cool.
Well, let's
and we will eventually talk about other viruses too, and then you'll see just, there's a lot more out there than just SARS-CoV-2.
I look forward to a time when it's no longer relevant to talk about SARS-CoV-2.
I do too, for many reasons. All right, let's do some email. Brianne, can you take that first one, which is anonymous?
Sure. Anonymous mentions that they live in East Tennessee and are a high school chemistry and biology teacher. And then they note that it isn't as glamorous a job as most people think. However, despite all this, I love my job. I love the kids. I love teaching science, and I keep coming back, although every year gets a little harder, and I dream of owning a small coffee shop surrounded by a lot of books. I totally understand everything there, Anonymous. I also love my job and the students and teaching science, but there are some places where it's hard. Anonymous says, I have to shop for my 90-year-old mother. I get everything from Trader Joe's right now, but she likes her Folgers coffee and Mayfield fudge bars. I went Wednesday to a grocery store, and it is Elderly Day, and I decided to wear a handkerchief. It is fun because I look like a gray-haired, middle-aged bandit. So I wore one shopping to protect the other elderly people in the store. When I got to the car, I took it off and put it in the bag to wash and then used hand sanitizer, which I despise, but alas, it is what it is right now. The handkerchief is doubled over, and I know it isn't PPE, but it has to be pretty good, right, for a 20-minute store trip? Probably. It's better than nothing, certainly. I'm curious why humidity and warmer temps slow down the coronavirus, if I understand correctly from the podcast last week. Is it just the natural cycle of them, prevalent in the northern hemisphere in the spring and have run its course by summer? It just seems humidity would keep the droplets in the air longer, wouldn't it? I know heat slows down microbial growth, but I wasn't sure how that applies to coronaviruses in general. We aren't seeing a lot of virus in our area. If you Google stats for Tennessee, these include large cities like Memphis and Nashville. Is there a chance we won't have a lot of cases? And if that is true, does that set us up for next year being a big issue for our area? If we get hit hard, our healthcare system, which is the pits as it is, won't be able to handle it. Could there be pockets of the population that do not get much exposure? And if this does happen, what could potentially be the consequences, good or bad? Yes. Well, Anonymous, some of this is related to what we were just talking about with the herd immunity in first and second waves. And so it is possible that if there aren't a lot of cases in your area, you could have a fair number of people who are susceptible in the future.
But the good consequence of that is you had fewer people infected and probably fewer people died.
Exactly. You have many more people who are healthy.
If you can prevent or mitigate that second wave, then you win. That's kind of the goal. Actually, I'm in Western Massachusetts, where we're kind of watching the eastern part of the state and saying, well, it's bad out there, but we get everything last. And in this case, that's a good thing. We've got currently our hospitals are not above capacity and things are just kind of smoldering along, and we're hoping that it stays that way and peters out a bit, and we'll just see.
Yeah, if you have these pockets of people who don't have much infection, then when the virus comes back in the fall or winter, then there will be other outbreaks. But as we've said, maybe we'll be better prepared. Maybe the hospitals will know how to respond better, and maybe there'll just be fewer infections. So,
and if the testing and contact tracing and quarantining runs fast enough, then you could just have little flare-ups here and there and say, okay, we'll tamp that down and just keep doing that until we have a vaccine.
Yep, the testing could detect it very early. The humidity and virus infection is a little bit more complicated because I think that there's something about length of time the droplets are in the air, but also how stable the droplets are and when they fall apart.
Yeah, so more humidity makes them drop faster from the air. She said stay in the air longer, but the opposite happens, right? They drop sooner.
Right. So they sort of get heavier with more liquid in them.
And then the heat, of course, would simply be inactivating infectivity or even drying out the droplets so that inactivates virus. Not really clear, but heat is part of it for sure.
And I don't know how much this applies at the scale of droplets, but certainly hot, humid air has a lot less lift. Aerodynamically, it's less dense.
Mm-hmm. I just want to say, and I'll say this multiple times in the next 6 months, when we do have these vaccines, please take them. Don't object. Don't tell me you're not taking it because they cause autism or something, okay? I want to hear it.
Oh, man.
Alan, can you take the next one?
Sure. Polly writes, I'm an American citizen, but I live in Vietnam and teach biology at an international school. And this is going to be a good summary of the Vietnam situation. Vietnam responded very quickly and closed schools immediately following the Tet holiday, so students have not been in school since the middle of January. As a private international school, we've continued to run school virtually, but local schools have been completely closed. For a long time, we had only 16 reported COVID-19 cases and all patients recovered. During this time, we were still allowed to go out and most businesses remained open. But within the last 3 weeks, there's been an increase in cases as people returned home to Vietnam from other countries. Now, probably because those other countries are closing all their stuff. Now, the airport is almost entirely shut down, and the government has closed most workplaces other than the essentials, such as grocery stores, gas stations, pharmacies, etc. Everyone is required by law to wear a mask in public or can be fined. Also, any building with an internal air conditioning system must turn it off and use fans instead, which is interesting. What I find interesting is the way the government has kept track of all the cases. They have a system where you're classified as F0, a person who tests positive with COVID-19, F1, person suspected of having COVID-19 or a close contact of F0, F2, person who's in close contact less than 2 meters for more than 15 minutes of an F1, and then F3 for further out, F4, I gather goes all the way out to F5, and attached a diagram, but it's in Vietnamese. Each level has a different quarantine procedure, from hospitalization to a quarantine camp to self-quarantine at home. Levels F0 to F3 are monitored by the local CDC. To me, it seems like they've really traced a lot of the cases accurately. I was even contacted as an F4 at one point because they thought I had been in an apartment building with a COVID-19 case, but it turned out I was in a different block of the apartment and so was cleared. Several people I know have been contacted and classified from F1 to F5. As of this week, Vietnam has about 230 confirmed COVID-19 cases and about 70,000 people officially in quarantine, including 300 U.S. citizens, according to the U.S. Embassy. 2 people I know were in quarantine camps in Ho Chi Minh City, where we live. The camps seem like they have varying degrees of comfort from videos and pictures I've seen, but seem livable. Easy for me to say, I wasn't actually there. My friends had basic Vietnamese meals delivered to their rooms, were provided with cleaning supplies and a mosquito net, had fans, and were allowed to go outside. They said the people working there were all very friendly. The biggest frustration was about how long the test results were taking. They were supposed to come back in 24 hours, but were taking more than 48. And if the test took too long, they had a second test and wait again. Unfortunately, I'm not sure what kind of testing they're doing. After 2 negative tests, both my friends have been released. Personally, I feel safe and am happy to be in Ho Chi Minh City rather than the U.S. right now.
Isn't that interesting?
Isn't that something?
Great. Isn't that something?
This sounds like
the benefits of a plan.
It's a plan.
They got a plan. Yeah, they
had a plan.
Why
and
can't
they're
we
executing
have a plan?
their plan.
Why can't we have a plan?
Yeah.
Yeah.
And it's annoying to them that the tests don't come back in 24 hours.
Yeah, we got people waiting for a week.
Yeah.
If you can get tested.
If you can get tested, yeah.
Rich, can you take the next one?
Michael writes, I'm writing to you from sunny Oahu, Hawaii. First, the local sit rep. 285 cases, 2 deaths with cases on all but 2 of the 7 inhabited islands. Oahu is the epicenter with 206 cases here. My guess is, I wonder if those are mostly in Honolulu. At any rate, we are beyond fortunate to have a practicing ER doctor as our wonderful lieutenant governor. Dr. Josh Green has been our local Tony Fauci, giving updates on the situation every day via his Instagram feed. He's been straightforward with us from the start while reassuring that we'll get through this together. He keeps a whiteboard that shows the nightmare projection curve over which he plots the actual cases per day. This shows how our social distancing efforts are flattening the curve. Here's a brief video of his update today, and he provides a link. The Center for Mathematical Modeling of Infectious Diseases at the London School of Hygiene and Tropical Medicine maintains a repository on GitHub where they publish models that they've been working on. Gives a link to that. On your last episode, an emailer asked, can we take number of deaths divided by assumed fatality ratio of 1% and estimate the number of cases? That's exactly what the team did in one of their models, only they adjusted deaths for the distribution over which it takes most patients to pass away. They can then compare the country's estimated range of actual cases to the number of confirmed cases to get a sense of what the percentage of cases in each country is actually diagnosing. Spoiler, most countries are under 20%. The U.S. is at around 16% at the time of writing, and Italy is just under 6%. This would put the U.S. at about 1.2 million cases right now, which certainly feels quite a bit more intuitive than the official 189,000 figure. At the other end, Germany is around 50%, South Korea is at 70%, and smaller outbreaks like Norway, Israel, Australia are estimated to be at or near 100%, which hints assumed fatality rate might actually be a bit high. It gives a link for those estimates. They also have fascinating plots of estimated effective reproductive rate by country over time, as well as a model specific to Italy. One more tool to plug, this interactive visualization shows the growth in cases and deaths by day and by country or state in the U.S./province in China using the Johns Hopkins data set. Please let me know next time any of you come to the islands. Your first Mai Tai is on me, Michael. And this letter is replete with useful links. So if you want any of these, you can go to the letters portion of TWiV and look them up. Thank you very much. This is very informative. This is really nice. And it's nice to know what's going on in Hawaii.
So your idea is right here. I mean, U.S. is about 16%. That's about what you estimated, right, Rich?
Well, yeah.
You were lower?
That's my hoping.
That's your hope.
Hoping that when we're done, it's more like if the case fatality rate, the infection fatality rate is on the low end of what the Oxford site predicts, then when this is all over, maybe we'd be as much as 30%.
Oh, right. These percentages in Michael's letter he's talking about the percentage of infections that we're successfully detecting.
Mm-hmm.
Right. Okay.
So the U.S. is estimated to, based on this model, to have only detected around 16% of our cases. In Italy, just under 6%. So
which gets you to something
1.2 million
anyway,
infections. Yeah, it gets you to about the same ballpark as what you're talking about.
Right.
I actually think it's even more. I think Ralph was saying about tenfold underestimated, and I buy that. I think this 1.2, it should be more like 1.9 or even 2 million cases in the U.S., but we'll see. Hopefully we'll see.
We could get the data. That's my point. We could get the data. Yeah. Let's get the data.
Microbe.tv/TWiV for the letters. These are all good links. Check them out. Mm-hmm. All right. Chaim writes on TWiV 596, Arthur in Oxford asked if people with SARS-1 antibodies might have some protection from SARS-2. Here's a preprint that screened 51 hybridomas from transgenic mice that were raised against SARS-1 spike. 4 were reactive with SARS-2 spike by ELISA. 1 was cross-neutralizing. Another preprint that addresses the reverse question. They studied 8 SARS-2 patients. Plasma from 7 of them cross-reacted with 1 on ELISA, but there was essentially no cross-neutralization. All right, so that's important because you could definitely find monoclonals that cross-react and even cross-neutralize. The question is whole serum, right?
Right.
Polyclonal serum. If that doesn't cross-react, then people who were infected not going to be protected. But, there are only 8,000 people infected with SARS-1, so wouldn't make much of a difference anyway. Continuing in the same episode, Trevor asked about return to work protocols. And the TWiV team discussed resolution of symptoms versus the cessation of infectivity. Here's a preprint that tested both PCR and live virus isolation. Sorry, Vincent. Their results suggested that, at least in mild cases, convalescent patients may no longer be infectious after about 10 days from symptom onset, despite continuing to be PCR positive. Preprint, medRxiv preprint for that. That's really good.
Yep.
Not surprising to me at all. Also not surprising, the live virus, because now we have all kinds of people talking about viruses and the live word. I mean, I have multiple meetings a day, and if I objected to every live, I would be pretty obnoxious. So,
this was an arc very early in TWiV's history. We had multiple episodes. You even had a survey, right? Asking whether viruses were alive
of course.
Or what.
Yeah, yeah. That's the old days when we did things like that. Anyway, Chaim, thank you
for those just joining us. The correct term for Vincent would be active virus versus inactive virus, rather than live versus killed or dead.
I would say infectious.
Or infectious. Okay.
Infectious. That's better.
Yeah, but people don't like it for some reason. I don't know why. I mean, I was on this morning on a 1.5-hour IBC, Institutional Biosafety Committee meeting, where multiple people use the L word. I'm going to call it the L word.
The L word. There you
go. I want to pronounce it. Brianne, you're next.
Yes. Dear TWiV, I wanted to share with you data from Taiwan. The discharge criteria used there is 3 consecutive negative tests, and the data is distributed between 8 to 53 days, with 24 being the median. Also today, 51 recovered patients in Korea tested positive again. People thought it might be an effect of faulty testing.
I don't know. So I guess these are PCR tests that Li Ping is talking about, right? Mm-hmm.
Yeah, I guess.
3 consecutive negatives. Well, that's more than other places.
That is. And imagine how many times you'd have to get tested and how many tests must be available if you could get 3 negatives.
I saw an article or a report on the TV about the Taiwan response to the epidemic the other day, and they have a plan, and they're on the ball. They're doing a good job.
And that was a letter from Li Ping.
Li Ping says, some scholars seem to suggest that chronic infection is a possibility. I don't think so. No. I mean, I think pieces of viral RNA will be shed for some time, but no, this is not a chronic virus infection. No.
And that's the thing to bear in mind with these PCR tests. They are very, very sensitive, but what they're detecting is just pieces of the virus. So if you test positive, you've got pieces of the virus in you, and that's a pretty good indication of, an infection that occurred there recently, but those pieces are going to persist a lot longer than the infectious virus.
Right. So this is just like with the previous letter, this is a difference between the infectious or AKA live virus versus virus pieces.
And in terms of people who get a negative test and then a positive test, there is some unreliability in the test, and because you're detecting at this low sensitivity threshold, you could fail to detect the RNA maybe even a few times in a row, and then the next time you do a test, you happen to detect the right little sample of blood or a sample of nasal swab that's got a little bit of RNA still.
Mm-hmm. Yeah.
I think of RNA hanging around after an infection as litter.
Litter. That's good.
Mm-hmm.
RNA is really a problem,
Yes, RNA is really
yeah.
a problem.
And if you think waiting 24 to 48 hours is a long time for a PCR, just think if you had to do a plaque assay.
Oh, yeah.
However, if it's a plaque, it's a plaque. If it's not a plaque, that's it. Oh, Alan, you're next.
Elizabeth writes to ask, has there been any report of increased sense of smell or taste as a symptom of COVID-19? I am recovering for what I believe to be a mild case of it, although I didn't qualify for testing, so I can't be sure. My sense of smell became significantly more acute. I could smell everything. Has this been observed in any COVID-19 patients you're aware of?
I'm not aware
I'm not aware of
Not
it.
that I know of, no.
But the loss of sense of smell doesn't seem to be a universal symptom either, so, everybody's body responds differently.
I suppose, loss of smell is, viruses messing with the olfactory epithelium. I don't know why it would increase. Maybe you got a mix of sensors there, and you get rid of some that are interfering, and the others work better. I don't know. That's why you get an increased sense. Yeah.
Okay, so Elizabeth continues, what was the reason for the second wave of the 1918 flu pandemic being worse in the fall than the first wave in the spring? That seems to be a popular example in the media for a virus getting more dangerous.
Oh, yes, it certainly is, and we can't escape it because we don't have all the genome sequences to show they're all the same, more or less. It's not the virus changing, and you would never prove it because we don't have all those waves of viruses. So to suggest that it is kind of a good position because nobody can prove you wrong.
Right.
However, the second wave is just, all those non-immune people, as we talked about earlier, right? Getting infected.
And that plus the natural seasonality of flu.
Yeah.
Right. So in the fall, this is flu season, and that's when you're going to get better spread of pandemic flu. Also, bear in mind, anytime you're reading about the 1918 flu, there was a war going on.
Yeah, that's right.
Yeah.
And so a lot of what was happening there with people moving around the world was related to the fact that Europe was basically on fire at the time. And there was, this horrific stuff going on, and we were taking huge numbers of young men and shacking them up in a barracks together and then transporting them on a ship across the ocean, then transporting them back across the ocean. And that changed in the course of 1918. Mm-hmm. So there's other stuff going on in 1918 besides the flu pandemic, and you have to realize that there's complexity overlaid on that. We should not read a whole lot into it. And as Vincent pointed out, we don't have sequences of all those isolates, so who knows?
Well, and you might imagine that some of the data on flu cases might have been slightly obscured by some of the things going on with the war as well.
Sure,
yes.
Totally. So Elizabeth writes, what was the reason for the second wave being worse? So that is a very loaded word, worse. It may be, I think you mean there are just more people, right? Doesn't mean that the disease was any different. I don't believe that it was.
And more people recorded as having died of the flu, as Brianne just pointed out, is not necessarily more people catching a deadly case of the flu. I mean, in the springtime, some of them probably got shot before they died of flu.
Right.
And if you look at the casualties from World War I mean, that was just thousands upon thousands of people, and maybe some of them had the virus and would have died of that, but they didn't have a chance because they got blown up.
But at any rate, Elizabeth, just wait for the next wave of SARS-CoV-2. We'll have all the genome sequences.
We'll get the data.
We'll have the data. And I will bet you a beverage of your choice there in Western Massachusetts. That's where Elizabeth's from, by the way. That they'll be very similar. The viruses, that is. Rich, you're next.
Amanda writes, I am the provincial co-lead for SARS-CoV-2 diagnostics in Saskatchewan, Canada. In Canadian public health labs, we rely heavily on lab-developed tests, especially in the molecular world. These do not need to be Health Canada approved. Because of this, as soon as SARS-CoV-2 reared its head, all the public health labs in Canada began developing their own assays. This is why we were all well situated to test early and often. Currently, in a province of only 1 million, we are running just over 800 tests per day and planning to ramp up. It shocks us that your provincial labs are not allowed to do this.
Yeah, it is shocking.
Yes,
well, it was. Now you can do it, but it's shocking that the CDC wanted to do it all in the beginning. It's crazy. Sorry.
No, that's fine. I believe I have some insight into the CDC kit issue. I won't name company names, but many of us at various labs across Canada received contaminated primers or probes at the beginning. For example, our RNA polymerase forward and reverse primers were contaminated with E template. Another lab had an E probe contaminated with RNase P template, and another with RNA polymerase template. This caused various effects depending on the contamination level. Based on what little they've said about their issues, it kind of sounds like they had the same problem. P.S. Don't hate on the millennials so much. At 36, I'm considered in that group, and all my friends are settled with kids and houses and sore backs and aren't out playing and spreading the virus. BPS. Vincent, someday I still expect a USB with your lectures on it so I can send to all my old colleagues in Papua New Guinea.
Were we hating on millennials?
I don't recall that, but we may have been like I was just talking about the UT kids
right.
who went off partying.
Okay, if they're in college now, they are not millennials. They are Gen Z.
Not millennials.
That's Generation Z.
Okay. Generation Z.
Yes.
As an almost millennial, I would say that no, they are definitely not millennials if they are currently in college.
It's actually interesting to look at the demographics of the infections that have been recorded in Austin, because the peak, at least last I looked, was in the age group from 20 to 30. And my guess is that those are the people that are out partying. I'll look that up again.
I had forgotten, Amanda, that years ago you asked for a USB with my lectures. So I just made a sticky. I'm sticking it right here because the course will be over in about 8 lectures, 4 weeks, and I will send it to you. Promise. All right, so the next one I love. Cheryl writes, will SARS-CoV-2 survive freezing, and for how long? You bet. That's how we keep it in labs. That's how we keep viruses. We freeze them. -70 is usually good, but -20 works too, depending on the virus. It can be years. Years. Vaccines are frozen. They can last for years. Why do you want to know, Cheryl? She didn't ask. She didn't say. But anyway, I just want to say you are all rock stars, she writes. You've always been. The rest of the world just noticed. Yeah. Isn't that great? She's right.
Thank you, Cheryl.
Haven't there been cases of like smallpox being thawed out from people who'd been buried in cold areas or something?
That's always been, people have talked about that as a possibility. I don't know that they've ever actually isolated infectious virus from a corpse. It's always been a story, an idea.
Okay.
Yeah, I just know about PCR.
Yeah.
Yes.
So I've just looked up the Texas data, and the peak of cases is in individuals in the 20 to 40 age group by a large margin.
So some Gen Z, some millennials.
Some millennials.
Brianne, you're next.
All right. Anonymous writes, hello, TWiV crew. Anonymous sends a link to an article in Science and says, I think this would be a great question for Brianne to address. I recently saw Bob Gallo dropping hints about an old vaccine on MSNBC, and I'm guessing this is what he's talking about. And so this link is about this question of whether the BCG vaccine, which is an old TB vaccine, it's been around since the early 1900s, might have some ability to be protective against COVID-19. There are actually 2 clinical trials currently going on, one in Australia and one in the Netherlands, looking at this very thing, and some interest in others. And it's sort of a interesting story that tests a lot of our basic immunology understanding. So the basic idea with vaccines is that vaccines rely on the adaptive immune system to make a specific response of B cells and T cells improved, so that when you're vaccinated, the next time you're infected, you have that nice specific response. There's some evidence that BCG, this tuberculosis vaccine that's given in some parts of the world, might lead to improvement in this new aspect of the immune system called trained immunity, which is sort of like memory responses made by the innate immune system. The innate immune system seems to be a little bit better at making certain types of cytokines. Once the innate immune cells are experienced, they might be a little bit better. And so maybe there's sort of this collateral effect against viral infection with this trained immunity coming from BCG.
So tell me if this is wrong. My understanding is that this was originally an epidemiological observation that in countries where there's more use of BCG, there appears to be less SARS-CoV-2. However, I would argue that in those countries, there's probably less surveillance as well.
I think I would agree with that. There have been a couple of studies not with SARS-CoV-2 that seemed to indicate that people who had some recent BCG vaccines might have had a slightly better response to different viral infections. Mm-hmm. It's harder to imagine what's going on with people who got BCG as children and how that is leading to really long-term protection. The only thing you could imagine is if there's less circulation amongst the kids in those populations, and that's helping the population. So I don't know that I really am buying the epidemiological studies about BCG in a population leading to protection many years later. But the boost to the innate immune system is possible. And that's, I think, why they are giving this to some healthcare workers in Australia to try to sort of increase their innate immune responses, their trained immunity responses right now before they see some COVID patients.
I think that's a really good experiment to do. The one caveat I would put on it is that BCG has been part of the standard vaccination protocol in China for decades.
Right.
So this is not going to be let's vaccinate everybody against BCG and then, SARS-CoV-2 goes away, because obviously that didn't work already.
Yeah, exactly. I think it has to be sort of a relatively recent vaccination that ramps up your innate immune system for, a somewhat shorter period of time. It's not going to be a thing where it protects you long term.
Mm-hmm. And it may also be not a huge effect, but if you're talking about healthcare workers who are getting a lot of exposure to constant, patient load, then this could make a difference.
Yeah, absolutely.
The next one from Ben is about the same issue, and so I think we could skip that and then go to Mark Allen.
Sure. Mark writes about coronaviruses and rhinoviruses. The common cold are the same, class 4 single-strand RNA positive, and I always thought our immunity to rhinoviruses only lasts a couple of years. but I've noticed in your podcast you expect coronavirus immunity to last close to a lifetime. Why would two viruses from the same class confer such different immunity durations? And then your podcasts have been the best thing on the internet for years. I'm so glad you're finally being recognized by a wider audience. Just a shame it took a pandemic to do it.
I think there are a lot of answers to that question.
Yes. So, just being in the same class of positive sense RNA viruses doesn't necessarily mean much, except that they're both positive sense RNA viruses.
That's a very broad class. A lot of
very broad class. It's
a lot of different structures, a lot of different members. So you can have viruses that really, in particular, in terms of what it looks like your immune system, look completely different. Okay.
Right. Humans, bats, and blue whales are all mammals, but we're very different.
Right.
And then the rhinovirus immunity, my understanding was that the reason you keep getting those is because we have so darn many serotypes of them, right?
Yeah, that's what I thought.
Yeah, I think that's correct. But, I learned a lot from John Udell last week. And so my view of coronavirus immunity has certainly evolved from the beginning of this outbreak, where I thought it would be durable, but even to the common cold coronaviruses, it's not. And you can get reinfected, but you just get milder disease. And so I would suspect that rhinos are in the same class if you really looked, but we were kind of distracted by the multi-serotype issue there. But, if you think about it, and the only respiratory viruses that give you durable immunity, which means lifetime immunity, are the ones that cause a viremia, like measles, like mumps, and like rubella. And they have a viremia stage. They go in the blood, whereas coronas and influenza and para-influenzas, etc., they don't. So maybe that's the connection. Maybe they all do a poor job because they never get to the blood. Maybe that's important. What do you think of that idea, Brianne?
Well, so as you were saying that, I was actually thinking, boy, I wish we were back in our discussion from immune, because I could think all sorts of ways about IgA versus IgG and whether you were making different types of antibodies when you had virus replication in the blood versus the lung. So I think that could certainly be a really important factor. And I'd like to think about that a little bit more, because I hadn't thought about that, but that's a really good point.
All right, Rich, you're next.
Nathan writes, do most disease curves for animal diseases often apply to humans and animals? Oh, most disease cures for animals' diseases often apply to humans and animals.
Well, we are animals, Nathan.
What diseases?
Yeah, I'm trying to think of examples.
I would say no to a first approximation.
So it depends what Nathan's asking. But the way I envision this is saying, do antivirals and vaccines against viral infections work the same way in humans and non-humans? In which case, yeah, right?
Okay. Yeah, and antibiotics are going to work the same in an animal and a human.
That's why I interpreted it, and that's why I stuck it in here, because I thought it would be an interesting thing to clarify. You make vaccines, right, for for cows and pigs and chickens to prevent them from getting virus infections.
Okay.
So that they all are working by blocking the virus from replicating, or by, in the case of antibiotics, blocking the bacterial replication. Sometimes we see big differences in terms of things like, the different types of trials that were done, like we heard about at the very beginning. So if you heard about some sort of antiviral that worked in animals, I wouldn't necessarily take it as a person. While the mechanism is the same, they're not necessarily interchangeable.
Right. That's what I was thinking when I said do a first approximation. No. So don't order veterinary drugs and take them.
No.
And there are huge differences in physiology between different animals and humans. Stuff that treats your dog's diseases could kill your cat and vice versa, and stuff that treats either of their diseases could kill you and vice versa. And so there's that whole aspect too, the physiology of it. But antibiotics and antivirals are a really good example of something that, if the same virus or the same bacterium infects a human or an animal, and you give the same drug, it's going to have a similar mechanism of action in both places, probably.
The next one's from Ati, who is an organ transplant surgeon in Dublin. He or she accidentally discovered the podcast and now is addicted to it. Excellent.
Great.
He or she is afraid they might turn into a virologist.
There's nothing to
be afraid of.
Nothing to be afraid of.
That's okay.
Well, I don't know. He's probably better off as an organ transplant.
Well,
surgeon.
yes.
Yeah.
We need those.
All right. Question. Why is COVID-19 hasn't been so bad in sub-Saharan Africa so far?
It hasn't?
I think there are not as many cases reported, but I would guess that is very much a reporting problem.
That's what I think.
That's
Yeah.
what I would.
Testing problem as
reaction.
opposed to anything else.
If you go to Johns Hopkins and you just go to the first page, all the big red dots everywhere else, they're not a lot in sub-Saharan Africa. They're a lot smaller and sparser. But I would bet that's reporting. Alan, you have said that before, actually.
Yeah, I think, and in fact, I asked this, I brought this up in a tweet weeks ago that no news is bad news in a disaster. We're not hearing anything out of places where we're pretty sure it must have been able to get to. So what's going on there? I think we have a letter much further down, and it's going to vary tremendously from country to country. Sub-Saharan Africa includes South Africa, which is, a pretty modern industrialized place, and also countries that are much more, much thinner infrastructure in healthcare. So I think there's going to be tremendous variability there. And just because you don't hear about cases in a particular country doesn't really mean a whole lot. I think we have a letter further down from somebody in Côte d'Ivoire where it actually sounded like they were doing a pretty good job handling it. But I don't think that's going to be the rule in that part of the world.
So the next one I think we should say for Daniel, because it's from a woman who's pregnant and wants to know the danger for the baby, because they're going to take the baby away from her for 2 weeks.
If she's positive.
Which she said, maybe I could get sleep.
Right.
Yeah, I liked that part.
So she wants to know, maybe we'll ask Daniel that. I don't know that. So that brings us, Brianne, to Robert.
All right. Robert writes, "Hi, you viral maniacs. If the selection pressure for the evolution of viruses is transmission, then viruses that are very good at transmission and that kill the hosts will end up with no hosts to infect. Thus, I would conclude that selection must be based on both transmission and host survival." I think that kind of makes sense, Robert. I think that the virus probably needs to balance transmission, but also have a host survive to transmit.
I agree. And I think a really good example is the use of a virus to try and kill rabbits in Australia, Rob, which is where you're from, right? The first year it was 99% lethal, and then both the rabbits and the virus evolved the next year to be much less lethal. So, yeah, if you kill all the hosts, it's not good.
Right. And there are still rabbits in Australia.
And so that's a great idea. Yes, unfortunately, that's a great example because, a few rabbits survived because probably they got randomly infected with a mutant that didn't kill them. And then the next year that virus just went crazy. So what do you think, Rich? Is that a good example of that?
Yeah. Oh, yeah. I think that's a great example. That is sort of the best large-scale example of virus evolution is the Australia experiment of introducing myxoma to kill the rabbits. And in the end, started off really lethal. And as you point out, the virus became, over time, attenuated and the rabbits became more resistant. So the idea that extreme lethality is not an evolutionary advantage is validated by that experiment.
I just talked about that very experiment today in class this morning. And we have a study abroad trip that goes to Australia. And the professor who runs that trip came to me last year and he said, I'm giving my students this lecture and it's in the Frank Fenner building of the or the Frank Fenner room of the Frank Fenner building of the Frank Fenner. Who's this Fenner guy? And I was awfully excited and made them take pictures and send them back to me.
Very cool. So I'm sure there are a number of listeners who don't know what we're talking about, this rabbit experiment, right? It's very cool. We've talked about it on a previous TWiV. You should look it up. Alan, can you take Vic's?
Vic writes, Dear all, this is Vic, currently unemployed cruise ship agent in Kristiansand, Norway. Sorry about your industry, Vic. My wife, Beate, Beate, who's an avid TWiV listener. Sorry I've mispronounced your name, probably.
Beate.
Beate. Thank you. Is an ENT surgeon at the Sørlandet Sykehus Southland Hospital here in Kristiansand. Last week, her hospital published 2 models that attempt to predict the coming peak of hospital admissions. I've attached that presentation. And then runs through the situation there in Kristiansand. The hospital is the only medical care facility for a stable population of 310,000 people. The nearest neighboring facilities are 2.5 hours away. All 310,000 are covered by health insurance and unemployment insurance, so there's no penalty for staying home. Consequently, they are staying put in their homes. The streets really are empty. All the daycare centers, educational institutions, gyms, sports clubs, barbers, beauty parlors, etc. are closed and have been since 13th of March. Basically, everything that would require more than 5 people to congregate is shut down. All Norwegian borders are closed. Travel out of the municipality of Kristiansand or across state borders is prohibited. Norwegians trust their government institutions and comply with the rules generally. Multi-generational households are rare. Even the very old have their own homes or apartments. The kids move away from home as soon as they are out of high school at 19. Given all the above, I want to move to Norway.
Yeah,
Yeah,
that would work.
It's not the first time I've thought that. Given all the above, this is an ideal population to test the limits of social distancing and makes the sort of modeling in the attached study as accurate as possible given what is currently known about the spread of SARS-CoV-2. As you can see from the charts, with an RE, R naught after measures that were taken in March, of 1.19, they're showing peak admissions on July 13th. With RE of 1.35, the peak is June 26th. Either way, a return to normal seems to be much farther in the future than the current American forecasts would indicate. My wife would like to know if you all think these scenarios are realistic. Can the R values really be that low, or is the hospital being overly optimistic? Well, so first of all, the R values certainly can be that low. And based on all you've said so far about the measures that are taken and the level of compliance, I'm not a modeler, but I would believe that if anybody can get the RE, the effective R value down into the 1-ish vicinity, that would be the kind of place where you could do it. So I'm not going to second guess the hospital's modelers. I think that's certainly a believable estimate. And as far as the return to normal being farther ahead than the American forecasts, well, bear in mind that in this country, we have this federalist system where basically every state does its own thing, and we have a national government that's been asleep at the stick since, quite a while before this. And so we've got all kinds of stuff going on in places that haven't closed down at all. And there's a lot more infection happening in many places. And it may be that many parts of this country see much bigger peaks that are much earlier. So that would explain that discrepancy.
It's possible for sure to get the R naught down, or the RE down to close to 1. That's how you stop an outbreak, right? But whether they're realistic or not, I don't know if they're predicated on what numbers of infections they are predicated on. Because as we've talked about before in this and other pods, we think we're underestimating them, right?
Now, I'm going to guess that Norway might be doing a better job testing than a lot of other places.
Maybe, yeah.
I know Iceland did an outstanding. I know Iceland's not Norway, but Iceland did an outstanding job because they've got a tiny population and a great healthcare system, and they just basically went around testing everybody. So based on what what we're reading here about the Norwegian situation, it's possible that their testing's a lot better.
I do think.
Would we also
go ahead?
Would we also expect that some of this difference might be related to when the virus was introduced to a particular location relative to when the social distancing started?
Mm-hmm. Mm-hmm. Yeah, I would agree.
Am I correct that Sweden is not taking these social distancing measures?
Sweden has just reversed course on this after they realized that their approach, which was initially the same approach the UK tried to take before they reversed course, maybe was not such a great idea. And I just saw a news article about that I think their prime minister came out and said, you know what? We really do need to do some more social distancing. And I'm not sure what all that entails at this point or how their outbreak is going to go from here.
By the way, go ahead.
Someone would argue that the Swedes, the Finns especially, are always
always social distancing, right?
Yeah.
So I looked up old Myxoma episodes. I found TWiV, and there are even older ones than this, I think. Oh, we had Grant McFadden on. I think he may have talked about it. At any rate, TWiV 547, the Red Queen meets the White Rabbit. And TWiV, I'm sorry, I said 547, 457, the Red Queen meets the White Rabbit. And TWiV 540, Wascally Wiwises.
Yes.
So together, those will tell the story.
I have a friend in Sweden, Agnes, who showed me around last year, and she said that they are upsizing the testing. She works in a microclinical lab, and she can do more than 1,000 tests a day. Biggest problem, shortage of reagents. So we have many backup platforms and methods. Everybody's very committed. So it sounds like, but she says otherwise Sweden is quite relaxed about the situation. So we don't feel that affected. Tell you more tomorrow. But she sent this picture that I'm going to paste in the show notes. She said we have toilet paper cakes, but not toilet paper. Picture of a baker with
weird.
Yeah, who would make toilet paper cakes? Thank you. I have to talk to Agnes today. Okay, that was Alan who read that. That brings us to Rich.
So am I at Elizabeth?
Elizabeth, yeah.
Elizabeth writes, Dear Twivians, kudos to you all, especially for Vincent's interview with Ian Lipkin. I have recommended the podcast so many times, I decided to make the recommendation part of my standard email signature.
Nice.
Good work. It was distressing to learn at the beginning of episode 596 that you have been recipients of impolite criticism. I am appalled that anyone who invests as little as 5 minutes in listening doesn't simply feel gratitude. Please pay no mind to such rudeness. Sincerely, Zabby/Elizabeth. Great.
Thank you. Thank you so much.
Thank you very much.
Thank you.
And she puts in her email footer, best source of scientific information about the novel coronavirus, TWiV, This Week in Virology.
Excellent.
Very nice. Aviva writes, thanks for all you do. My social media just alerted me that HEPA filters are made of tiny glass threads. If this is true, please don't cut them and breathe that in. Here's the PSA I saw. So my wife has been cutting up HEPA filters. So I went and looked at, they look like they're full of glass fibers. I don't know if that's a problem. Do you, Alan?
I don't. I know what HEPA filters are, but I'd never thought about what they were made of, and I'd certainly never thought about cutting one up.
Well, Dixon was talking on Friday about they're making cloth masks, and they're putting the HEPA filter in between layers of cloth. So I think that's probably fine because the glass fibers are not going to go through the cloth, right? But I reserve, I'm not recommended. If anyone has more, I don't trust this particular video because it's, a excited YouTube video and who knows. So, all right, let's do one more round. Okay, folks?
Cool.
Okay.
Brianne.
Sure. Allison writes, the coronavirus info is necessary and great, but I do look forward to when things get back to normal and y'all can discuss papers again. I kind of agree, Allison. Here in North Georgia, it is a beautiful 75 degrees Fahrenheit and sunny. We have enough pollen to choke a horse, though. Remind me not to visit Allison. So I have a question about making masks at home. There have been patterns floating around where you take vacuum cleaner bags and sew them into a mask. The idea being that the vacuum bag will filter the air out. I myself am not really familiar with vacuum bags and how well this would or wouldn't work. Do you have any thoughts? So strangely enough, I have received some other emails on this front and actually found a paper that talked about some of this.
Oh, cool.
And looked at the efficacy of different household objects as masks against influenza.
That's great.
Like I said, I strangely enough got this question last week. And vacuum bags were number 1 in their study. While kitchen towels came in number 2. So I know only the information from that particular study, where they looked at things like linen, scarves, kitchen towels, pillowcases, vacuum cleaner bags. And yeah, the vacuum cleaner bag and the kitchen towel worked the best, but were not easily wearable.
Right.
I think
and presumably, if you're using a HEPA filter bag, then maybe look closely at what you're cutting up or not put it directly in contact with your face. And this all presumes we're talking about clean vacuum cleaner bags, of course.
Yes, absolutely. I just put the link in.
Just wanted to point that out.
I think in all cases, you should sandwich them between cloth just to be sure, right?
Sure.
Yes,
I think that
absolutely.
makes sense. That's very interesting. Yes, we have gotten tons of face mask email here at TWiV all about making them and this and that and the other. It's amazing how it's exploded. But as you know, we have been told we should be wearing them now. So
this is a great find, Brianne. Cool.
Yes. I guess we should thank some of the people who are sending me really interesting questions by email.
Yes.
For sure. Alan?
Leonid writes, I'm writing because on a recent TWiV, a listener asked about the difference in the public health response in South Korea and the U.S. It seems to me the question should be posed more broadly, as this is a difference not between South Korea and the U.S., but between South Korea, Singapore, Hong Kong, Taiwan, and Japan on one hand, and the U.S., Italy, Spain, France, UK, Germany, etc. on the other. I think there are 3 components to the answer. First, expert in media opinion. Well, first of all, I just want to comment on this categorization of countries. I think that the Asian group may be a little too broad. I gather Japan's response was very different than that of South Korea's, for example. And the European group is probably also too broad. Germany's response was quite different from the UK or Italy. Anyway, continuing with the letter. First, expert in media opinion at the outset, countries in East Asia have had to face these emerging viruses seemingly every few years. So saying that there was a lot of coverage and expert discussion in those countries as SARS-CoV-2 came out. Second, staggering incompetence at the CDC, apparently at many levels. Thankfully, Dr. Fauci rose up to the task. Perhaps he will agree to clean up the CDC when this is all over.
Cleaning up the CDC is not part of Fauci's job description. That has to come.
No.
That has to come from somewhere else, but it needs to be done.
Yeah, and I would point out that there was a change in management at the CDC just a relatively short time ago, and that is a big part of this. I don't think it's certainly not the director of NIAID's job to clean up what's going on at the CDC. And I am pretty sure that Tony Fauci, who has been invited by multiple presidents to take a promotion and direct another institute, is not going to take over the CDC. He's at NIAID. That is what he does. And we love him.
And thank goodness he's there.
And thank goodness he is there. He's exactly where he belongs. Third, cultural differences. I doubt that many students in East Asia would be found enjoying their spring break on a crowded beach as an epidemic is sweeping across the country. A friend in New York tells me there are still a lot of people on the street. I do not see such carefree and self-centered attitude as a problem in and of itself, but it's very harmful at times like these. I hope the above can be remedied. After all, SARS-CoV-2 is not even the worst that can hit us.
That's for sure. Actually, New York is pretty deserted, at least up by the medical center. I just wanted to say that I visited Singapore last December, and I visited the CDC, and they are still remembering the Nipah outbreak, which happened partially in Singapore, and they have built an infrastructure to prevent that sort of thing from happening. They are very aware of the threat of emerging infections, and they have really taken it seriously and built plans and infrastructure and so forth. I mean, I haven't published any of those TWiVs, but one of them, they have a whole division just making Wolbachia modified mosquitoes for release. It's amazing. The CDC that they have is a combination lab and hospital. And, the director who I spoke with, she was in the middle of Nipah. And so I think because of that experience, they're more ready than we are because it's too far away from us. It's really good points.
I want to go back a minute just for a moment to the previous letter. I forgot to comment. Allison was commenting, it'd be nice to get back to the normal show. And it occurs to me we probably got a lot of listeners now who have never heard the normal show. So
probably true.
When this is over, hang in there. There's a lot more virology than just coronavirology.
And if you want a preview of what that's like, you can go back to some of the old episodes. They are all online.
Which is a good segue into the other thing I was going to mention, and that is, I know we've mentioned this since the pandemic started, but we interviewed Tony Fauci in 2013. That's TWiV 219, Fauci Pharmacy. And my guess is that you can't find an hour-long interview with Tony Fauci anywhere. But there it is.
Yeah, we do long form here on TWiV. Please don't leave after the pandemic. You really just listen to some other virology. It's really cool.
I actually had a family member recently who asked me, wait a minute, could there be another virus after SARS-CoV-2?
Oh, boy.
And I responded, yes, that was entirely possible. So you should continue listening so that you can know more about those viruses and all the other ones that are out there.
I get the daily email from ProMed Mail, and I can tell you that SARS-CoV-2 is not the only virus causing outbreaks right now. You just don't hear about the other ones.
Mm-hmm.
Rich, can you take Greg's?
Greg writes, I was wondering how Dr. Lipkin is faring this week. Can you answer that, Vincent?
He's very well.
Good.
Unfortunately, a lab member of his is now sick, but he's much younger and is getting well also.
Let's see here. And then he says, second, weren't you all just thinking about this SARS-CoV-2 in wastewater, and gives a link to a Lancet article? Is this the one in Netherlands? Yes, we did cover this. Yeah. That we were talking about looking for SARS RNA or even virus in wastewater, and in fact, that's been done. So we linked that paper last time as well. Third, I saw the I saw the following commentary in the Lancet by Gregory Pollard. It echoes what Dr. Lipkin and others have recommended regarding a nationwide lockdown being a prudent route to take. Unfortunately, there seems to be a lot of political and not to be a lot of political and societal will for such a wide-ranging move. Yeah.
I love it. Greg is a PhD candidate in English writing, rhetorics, and literacies. There you go.
Excellent.
Pandemic will attract everyone to viruses. Okay.
Although we had a pretty diverse audience even before the pandemic.
Truly. Okay, the last one is
and
from
actually, Gregory, hang on, because we get into crazy arguments about writing, rhetoric,
yes.
and literacy.
Yes, we do.
Yes.
That you could probably respond to.
We may need your expertise in the future.
Exactly.
Yes. Yeah, weigh in for sure. Yeah, we're into this pedanticism.
Yeah.
All right, our last one is from Colin, who is appropriately a PhD student in Singapore. at the Duke National University of Singapore Medical School, which I visited and did a TWiV there. Very nice visit. I don't know if I met you, Colin. He writes, "Dear, this week in COVID-19 hosts. Situation in Singapore. While we initially seem to have COVID-19 under control, the last few weeks have seen a rise in the number of locally transmitted cases and new clusters despite gradual implementation of various social distancing and work from home measures. As of today, 4th April, we have 6 deaths. Yesterday, the government decided to act more aggressively and is implementing a lockdown starting Tuesday, which is today. Essential services will continue. Markets will be open. Non-essential workplaces shut. There's a distribution exercise starting next week whereby reusable masks can be collected by residents. You wash them after each day, apparently. While there are valid criticisms of how China has handled the outbreak, there are many lessons that can be learned from the measures they've taken." And he provides a link to an article that shows how 16 shelter hospitals, over 10,000 beds, were set up in existing stadiums, exhibition centers in and around Wuhan in February. Well,
I just wanted to back up to a sentence that I neglected to highlight the first time through. He says, crucially, in my opinion, the government has evidently been planning for this implementation for weeks, talking about the shutdown.
Yeah.
A plan.
Yes.
If you get a theme here, it's that we don't have a plan and we should have been responding back in January.
And now there is no excuse whatsoever for not coming up with a plan, because this is only the beginning. You know, we may get a breather having socially distanced ourselves, but we don't know what's coming down the road. It would be nice to have a plan to deal with whatever it is.
Yes.
All right. That is this midweek TWiV. We're doing number 600 on Friday, folks. Wow.
Yeah.
And I think
Yay.
everyone is going to be there, which is great. And we'll do it at our regular Friday time, which is what we've been doing for the past 12 years. You can find all the show notes in these letters at microbe.tv/TWiV. Send your questions and comments to twiv@microbe.tv. And consider supporting us financially, microbe.tv/contribute. Brianne Barker's over at Drew University on Twitter. She is BioProf Barker. Thank you, Brianne.
Thanks. It was great to be here and get a chance to talk about all of this.
I'm glad you could make it. I didn't think you were, but very happy to have you along. Rich Condit is an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove's at turbidplaque.com on Twitter. He is Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.blog. I want to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.